Okay, the first lab that's on this exam is the plant lab. I told you when we did that lab that I'd like you to be able to look at the cross-section of a monocot root, monocot stem, eudicot root, eudicot stem, and know which is which without looking at the label on the slide. And so I'll cover those labels. I've got all four of those set up at the far right over there. You have a handout that goes with that, and there's also most of that information is in your lab book. So be able to identify. You're just going to see a cross section of a stem or a root. You have to tell me, is it a stem or a root, monocot or eudicot? There'll be a couple of those on there. There are four different possibilities. I won't use all four of them, but I'll use a few of them. There's also from that lab a slide of tilia, and there's 3 growth rings on the one I have set up there. If you don't see 3 growth rings, you can ask me to come over and help you identify them. But that would be considered a 3-year-old stem, because we're just counting the growth rings here. In lecture, I said some different stuff about that, but just count the growth rings. Also, the root model, that's a modified one. It's not the same one that was out before. That one has a key that goes with it. Know the zones of cell division, elongation, and maturation. Know where the ground tissue, the vascular tissue, and the dermal tissue are. That's it for plants. The flower model and leaf model, we're done with those, so don't worry about those anymore. As far as cnidarians and poriferans, so sponges and cnidarians go, when you see any sponge specimen, all you need to know is its phylum, that it's in phylum Porifera. And if the stovepipe type sponge, I held one up and showed you the opening at the top is called the osculum. The cavity inside that opening is called the spongiocele. Spongiocele and osculum are both potential test questions. If you see, and then also for the sponges, we have a slide of sponge spicules. You should know they're made of silica and be able to identify them. There's also spongin fibers, sponge skeleton, so on. I've got the yellowish one on the slides over there. I think that's it for the sponges. For cnidarians, you should know all the specimens. If you see the jellyfish Aurelia in a plastic block or a jar, know that's in class Scyphozoa. Know also that anything in the jellyfish form is in the medusa form. We even have a medusa form for the Obelia hydrozoan. We've got the Hydra slide over there, one of them, and we also have the Obelia slide, which is a branched polyp. So both the Hydra and the Obelia colony or colonial hydroid, those are all polyps, either single stalk or branched polyps. Then we have the Obelia medusae. These are little tiny jellyfish forms. And then the other body form you see in the cnidarians is the planula larva. And we have that slide as well. And you should know that's a young form of some cnidarian. That particular one is for a jellyfish, but it could be for Obelia or another hydrozoan. Nematocyst slide is part of this, and you should know that when you see it. You should also know that that sponges have no tissues. They have no tissues at all. That cnidarians have 2 tissue layers, and the ones that they have are endo and ectoderm. The one that they're missing is mesoderm. I'm going to ask you either which ones do they have or which one don't they have. Okay, and then there's probably a couple jars or so on. Just know their name and their class. I may come back to that, but I'm going to move on now and talk a little bit about flatworms. And any planarian you see, you should know belongs in phylum Platyhelminthes, and it's also in subphylum Turbellaria. And I mentioned a slide when we were setting up that lab that had 2 planaria in it, and one was pink with the black stained GVC. That's one's featured on those scopes, because I want to ask you, what is that black stained structure inside? That's the gastrovascular cavity. And that's the whole mouth slide, and that's prepared over there as well. If you see any tapeworm, know it's in phylum Platyhelminthes. The subgroup it's in is called Circomeramorpha. and then know that its most advanced organ system is the reproductive system, and that the other systems are greatly reduced because they don't need them. So the reproductive system is, you see a tapeworm slide, what organ system most advanced? Please pick reproductive. Let's see here. Okay, I guess I didn't really mention it, but the Hydra and Lobelia, you should know the class they're in. They're both in Hydrozoa. They're both in the polyp body form. The Medusa would be the slide of, or the jar, I should say, of the Aurelia. Okay. You should remember the spongin fibers are made of the protein spongin. That's why they're called what they're called. That's the commercial sponge fibers for the sponge skeleton. 3 different names for the same thing. The sponge skeleton is made of the protein spongin. And that planaria or planula larva is motile to help disperse the offspring. Okay. So we'll talk a little bit more now about the other stuff in this lab. So we have the roundworms. When you see, we have 2 slides over there. One's a cross-section of a male, one's a female. I didn't identify them, but the larger one is the female, the smaller one is the male. You can see the frilly muscle inside the outer body wall, but no muscle around their digestive tract, which is sometimes hard to find. On the female, the larger one, I put the pointer on the digestive system because it was a little hard to find. But there's no muscle around that. So that's a pseudocoelom. It'll be in the slide. You'll need to know they have a pseudocoelom. That's the only phylum that does. Now, a slide I didn't talk about last week was the trichinella slide. What we're seeing is in that slide, skeletal muscle. The trichinella worm is a parasite that's found in muscle. And if you eat uncooked muscle, it will get into the gut, and then the worm can burrow its way into the muscle of the new host, reproduce, and infect that muscle. And then if somebody eats that muscle. Now, if you cook the muscle, it's not going to be a problem. It'll kill the trichinella worm. But what that slide shows is some striated or striped tissue, that's the muscle tissue, and then little circles in it are the trichinella parasite. And the animals that are most associated with trichinella are pigs, because they eat dead pigs. They're omnivores. They'll eat meat and vegetable material. And of course, pigs don't cook their food before they eat it. And so you shouldn't eat raw pork. Now, some people I see on networks like the HGTV or something that talk about food say, oh, there's no more trichinella in our pork supply, so that means that it's okay to eat raw pork. And I don't buy that because if you got to trust that if the farmer is not going to feed the infected pig to the living ones. And I don't think trust is something very common nowadays. Somebody tell me why you couldn't get this in cow. Why couldn't raw steak have trichinella?
Cows are herbivores.
That's right. They don't eat meat, so they can't get infected by something that's in meat. So that's why it's okay to eat like steak tartare and rare steak and things like that. Okay. So know it's in muscle tissue and it's trichinella, and the slide will be there for you. For any bivalves, any clam that you see, whether it's the Musselglochida slide or just a clam in a jar or a dissected clam, you need to know they're in class Bivalvia, phylum Mollusca. Remember the gills in the bivalve mollusks are used to do gas exchange. They're also used to catch food because the water with food in it passes by the gill and the food sticks to the surface of the gill, and then it gets swept to the mouth where it's swallowed. So gills do both gas exchange and they help with feeding in a bivalve mollusk. If you see a squid or an octopus, you need to remember that it's in class Cephalopoda, phylum Mollusca. About these, I would ask whether they have an open or closed circulatory system, and if they're octopus or squid, they have a closed system because they move more, unlike their other mollusk cousins who don't move much and have an open circulatory system. Earthworm, got the cross section over there. I put that slide up and I put the pointer on the typhlosaur. That's that structure that increases the surface area for the absorption of nutrients. So the typhlosaur is involved in absorbing nutrients. I've got the parapodium for the polychaete worm that's called Nereis, and that's the one that they use for paddling, so it's going to help for movement or locomotion. It's also got blood vessels inside and surface area outside to exchange gases, so it's involved in gas exchange. And it has the sharp setae. On the slide you can see these black slashes with a point at the end. They're going to stab any predator that tries to eat them. And so that's defense. So remember, the parapodia are functioning in locomotion, gas exchange, and defense. That's very much on the test. There's a leech slide there. When you look at the leech slide, the one thing I want you to know is which is the head end and which is the tail end. You can get that information by looking at the lab manual. There's a sucker at each end to help it hold on to the host. And so there's a bigger one at one end and a smaller one at the other end. So which end has the bigger one and smaller end one? Again, that's in your lab manual. The bigger one, well, it doesn't matter, but one's called posterior, one's called anterior. Anterior at the head end, posterior at the tail end. I'd like you to tell me the difference between the two. The slide has kind of both of them barely fitting in the field of view because it's such a big specimen, you can't get the whole thing in the field of view. Let's see. Okay. For the cross sections of the stems and or roots, the eudicot stem and the eudicot root, monocot stem, monocot root, for all those cross sections, you should know where the vascular tissue is, where the dermal tissue is. That's always the outside of the ring, so that's easy. Vascular tissue typically has some shape that helps you identify it. And then everything in between is ground tissue. On one of those, I might put the pointer on one of those three tissues and ask you which one it is. Okay, if you see any snail or slug, know it's in class Gastropoda, phylum Mollusca. For the nematodes, we have some Ascaris parasitic worms in plastic. And I don't know if I mentioned to you guys, I know I mentioned it to the afternoon class, but in those plastic blocks, there's two worms, a longer one with a straight tail, that's the female. The shorter one with the hooked tail is the male. So I'm gonna ask you, I'm gonna put a sticker on that plastic block on either the male or female and ask you whether it's male or female. And the one with the hook on the tail is male, and the one with the straight tail is female. Couple dissection questions for the clam. Remember the valve or shell, the mantle, the foot, the gill, and the adductor muscles. I'll probably have at most 2 clams on there, asking about 4 of those 5 things. And then for the earthworm, probably only 1 earthworm, but I might ask the septum, crop, gizzard, intestine, clitellum if there is one. The ones I mentioned in lab, but I'll probably have 1 earthworm and ask about 2 of those things. Okay, there's also a chitin, a mollusk that's in class Polyplacophora. You might be asked about that. And if you see any earthworm dissected or in a jar, know that that's in class Oligochaeta, phylum Annelida. Okay, I think that's pretty much it. I think I've covered just about every question. So you're all set now if you want to do any studying or ask any questions. I'll stick around until